Okay, well, welcome everyone. It's always a pleasure to be invited by students to give presentations, so that's nice. This reminds me a little bit of COVID times, because last year, I must say, I've been giving quite a lot of presentations, but face-to-face, which is, of course, the nicest way to do it, I think, but webinars are also very nice and convenient for people to follow and attend. So I'm going to speak about the work that we are doing in Utrecht in my group, which is about well, solving 3D puzzles. So we are trying to model interactions between biomolecules at the atomistic level. So I'm going to give you a short introduction on the topic and then explain you how we are doing this kind of modeling using Haddock, a software that we have been developing for more than 20 years now in my group. I'm going to show you the new directions that we are heading to in a new version of the software. And then I'm going to go into some application examples. One, which is the modeling of antibody-antigen complexes using AI these days, of course, to generate initial models. And the second example will be an application of small molecule docking using Haddock and some new capabilities that we have. And I will finish with some general conclusions. So interactions are everywhere in life. So what you are seeing here is the center of Utrecht, which is an old city with an old university. It's a beautiful city, lots of old buildings, and the river that goes through the center of the city is the Old Rhine. And what you see a lot of terraces there, and at these terraces you see a lot of interactions between people at the macroscopic level. But everything in our cells and also communication between our cells is controlled by interaction at the molecular atomistic level between molecules, and in particular proteins are playing an important role in this. So if we want to understand the social life of cells, we need to understand how molecules talk to each other. And for this, we need to take the step to understanding their 3D structures and the 3D structure of the complexes. So we have the genome, which is basically the genetic information coding for the protein. So when a genome comes to expression, so we get the set of proteins. And then these proteins start interacting with other molecules or other proteins, and they form the next level of organization, which we'll call interactome. So what you are seeing here is such an interactome where each dot represents a protein and each connection represents interactions between proteins. And if you look at these, there are a lot of data out there. Some of these data are coming from high throughput experimental techniques. Some might be also predicted. But if you do an analysis, general analysis, you will realize that there are many more connections than there are dots. So proteins interact with many partners during their lifetime. So in a human proteome, we have in the order of 21 in human, we have about 20,000 proteins that come to expression. So our genome encode for 20,000 proteins, but these will form several 100,000 interactions with our biomolecule. So the complexity of this network of life is much larger than the complexity of just the player in this network that are the individual proteins. And as in life, miscommunication leads usually to problems between people. Miscommunication in those networks here also lead to problem, which will be the cause of disease. So understanding this communication network is important also to develop new drugs, biologics, and interfere with the network when it doesn't work properly. So this brings me to the structural biology of interaction. So how do we study those complexes at molecular atomistic level? So what you see on the right side of the slide are these experimental methods that are giving you access to basically structures or parts of structures of biomolecule. And the oldest one is X-ray crystallography, and then we have NMR and cryo-electron microscopy. Those three are the classical methods these days that allow you to get really atomistic information about structures of biomolecules. But next to those, there are many other methods, and this is just a small list. There are many other ways of getting information. So you have mass spectrometry, for example, is very important these days in generating bits of information. So it will not allow you to solve fully the structure of the complex, but it will give you pieces of the puzzle. And you will have to assemble these pieces using computational methods. And then there are also methods. It can be as simple as doing mutations in a protein, and some assay that you run a gel and see if a complex is formed or not. So all these experimental methods are going to give you pieces of information. Alone, they will not allow you to solve the 3D structure of the molecule you're interested in, but in combination and together with the computational methods that are now on the left side, we can generate 3D models of biomolecular machines. So historically, there's a number of different techniques that you can use to build 3D models. Many of those are based on molecular dynamics simulations, where we describe the interactions between particles, between atoms to build model. You can try to model by homology, but the topic for today will be mainly docking. But of course, since several years now, there has been a big revolution in, say, structural biology, computational structural biology, with the advent of AlphaFold, developed by DeepMind. And very early on, when AlphaFold was published, initially to predict structure of single proteins, people realized that you can also use it to predict structure of complexes. And there has been a lot of work, and these are just a few publications, and the latest publication, which is making a lot of buzz these days, is AlphaFold version 3, which shows improved performance, but also allows to model protein, nucleic acids, glycan, different types of molecule. AlphaFold 2 was limited purely to proteins, and now with AlphaFold 3, the field has widened since we can now target other types of molecules. So AI is really playing an important role in everything that's happening these days in terms of structural biology. Still, for today, we will go back to the more classical approaches that have like molecular docking. Molecular docking is about probably 50 years old by now. And the simple concept is given two molecules, but you also have to realize that many complexes, many interactions in our cells are made of more than two molecules. So we need to be able to model large assemblies consisting of more than two molecules. But classically, docking is associated with, say, a binary problem. So given the structures of the constituent of your complex, can you predict how they are assembled in space? For this, you need to basically sample all possible rotations. So you can fix one molecule in space. And for the second molecule, what you have to do is translate it on the first one and rotate it. And for each rotation that you sample, you have to translate it. So you're going to generate in this way a lot of models. And then you have to assess the quality of the model to try to predict which one is the correct solution. For this, you can use shape surface complementarity of the molecule. You can use classical physics, like using interactions between charged particles, electrostatic potential, shown here on the top, van der Waals interactions. So these are basically classical energetic description of the molecule and the interactions. Of course, you can also use machine learning AI these days to discriminate all of that. But the concept is basically I sample a lot of possible assemblies, those molecules. Two phases. One is the sampling. So here you have some kind of simple energy landscape, which we don't know a priori, but we need basically to search through this energy landscape. So, and in the case of complexes, we will call it interaction landscape. So you need a method that samples this landscape. That's the sampling problem. And you need a scoring method that basically is going to associate some score, energy, whatever you want to call it, to all the model that you generate. And the hope is that the model with the best score, the lowest energy, will correspond to the correct solution of the problem. Now, you can do this exercise purely based on purely ab initio, so not using experimental data. But if you have data that are coming from experiments, that are coming maybe from bioinformatics analysis, you can decide to use the data in several ways. So you can use the data to bias the sampling, where you say, well, I don't need to sample the entire space, which is possible for those two molecules, but I can limit my sampling to the relevant space for the data that I have at hand. And you can also use the data to score the models that you have, because you want your models to fulfill or to explain the data that you have measured. And this distinguishes basically different approaches in the field. So most of the initial docking methods were basically doing this global search where they were systematically searching all translation, all rotations. So you have to search a large space. And if you have data, and this is more the era of integrative modeling, information-driven search, you can focus the search on a small region of space, spending more time maybe to get better models here. So both methods have advantages and disadvantages. So here you spend more time maybe searching the entire space, and the space is really large. Here you focus the search, but if you put bad data into this process, you're never going to find the right solution because you're searching in the wrong place. So there are advantages to going more systematic and sampling every possible solutions, and there are also advantages to using data when they are available. And HADOC, the approach that I'm going to describe you, is basically following this information-driven search approach. So let's go to HADOC. So HADOC has been developed for more than 20 years by now. The original publication was in 2003. And what triggered basically at the time the development of the software was that there was not so many software that allow you to model complexes. Docking was already there, but docking was functioning purely in an ab initio mode without incorporating any information. As I'm embedded in an experimental institute, we had access to a lot of data. And at the time, NMR data that are pinpointing where the binding surfaces are on the proteins. And this was the trigger basically to develop a method where we could use this data to really drive the modeling process. So it started from NMR, but very quickly it evolved in using mutagenesis data, biochemical data, cross-linking, HD exchange. These are kind of data that you analyze typically by mass spectrometry. We went into bioinformatics, so trying to predict where things are binding from sequence. NMR can give you information about orientation of molecules. And then you have all the methods that are giving you shape information. In 2000, in the year 2000, cryo-electron microscopy was not yet established as a method that will give you atomistic resolution. Now, these days, so there has been a revolution in cryo-EM where you can get really atomistic information on the system, at the time not. But there are other methods like small angle X-ray scattering, which is still giving you shape information, but not at the resolution that you want to really build a 3D model. But this is still information that you can use in this modeling process. What distinguished HADOC also from most of the docking software that were out there at the time was our ability to deal with more than 2 molecules. And this was implemented pretty much at the start of the development of HADOC. So currently we can go up to 20 molecules. It's kind of an arbitrary number. But of course, the interaction landscape that you have to sample when you increase the number of molecules that you are dealing with becomes huge. And it really only makes sense to model very large system in this way if you have good information to drive the modeling process. HADOC also allows flexibility, so we are not dealing with rigid puzzle pieces, but we're going to adapt the shape of the puzzle pieces during the process. What kind of molecule we support? So that's also something that very early on in the development of HADOC, we had support for different types of molecules. So we started working with proteins, peptides. Very quickly, also early on, we started dealing with RNA, DNA, nucleic acids, glycans, also very important. I think the importance of glycan has been clearly underscored with all the COVID pandemic, where you've seen the spike protein decorated by all those glycans. So this is a topic we are still working on actively. Small molecules, so you can also do docking with small molecule docking with HADOC. We have taken steps to go to larger system by simplifying the representation of the system. Instead of going to a full atomistic description, we simplify by using what is called coarse graining. And we have also the ability of defining arbitrary shape, which we can use as restraints. So you see here such a shape, 2 planes, and this could represent a very simple representation of a membrane, which you could use to position proteins inside, in between the 2 planes defined by those bits. So this is a type of system, so we can mix and match different types of molecule, and we can mix and match also different types of information of restraints. So anything that you can translate into distance is really native to HADOC. but you could also define rotations, diagonal angles. You can use orientational information from NMR. You can use cryo-electron microscopy densities. If you have not reached the resolution at which you can really model at atomistic level, you can still use these densities together then with ad hoc as a way of building complexes. Symmetry, if you have to generate symmetrical molecules, homodimers, trimers, tetramers, you can impose symmetry also in a modeling process. Now, how do we do all this modeling? So ad hoc consists of 3 stages. So in the first stages, we're going to treat the molecule as rock solid, meaning by that the only degrees of freedom or motion that are allowed are the overall translation and rotation of the molecule. So this is illustrated here. So what you see here is basically a molecular docking of 2 proteins, which is driven by NMR data in this case. So the spheres that you see are the residues that were identified by NMR as being important for the binding. So this is a very fast process. So we can sample here thousands, tens of thousands of models. And then we need to go to the next stage, is to optimize those interfaces, because at this stage, the molecule are rigid, so there is no conformational changes. But proteins are dynamical, and dynamics is often an important part of their function, also in terms of molecular recognition. So the second phase in ad hoc is to take a few hundred of the model that we generate by rigid body docking, and then start optimizing the interface between the molecule. And if the movie comes over fine enough, you will see that the side chain at the interface of those complexes start to move. So we are now doing molecular dynamic simulation, so simulating the evolution of the system as function of time, given the energy function that describe all the interactions. And in this way, we can introduce small conformational changes between the molecules. That's a refinement stage of ad hoc. Now, the final stage used to be to take the solution that we have, put them in water. So we surrender the complex with a layer of water and run again some short molecular dynamic simulations. These days, we don't do that anymore. We just do a short minimization because it's faster and it doesn't change really much the quality of the models that we are getting. So we generate, say, 10,000 of models in the initial stage. We refine a few hundreds. And then we have to decide what are the good models and what are the bad models. So that's the scoring problem. So ad hoc is still following a very classical scoring approach using classical energy functions. And at different stage through the protocol, we might use a different combination, but what you are seeing here is a simple linear model that combines energies into a score. And we use this score to rank the model that we generate. These days, you should say, are we doing machine learning to do scoring? A linear model is a machine learning model in principle. So what's in there? So how do we score the models that we have at the end? So you see 2 terms that are indicated in green here. So the first one is the electrostatic energy, but we only calculate for the scoring the energy between the molecules. And the second term is the van der Waals interaction. So again, this is the amount of energy between the molecule. So the sum of the 2, and we use 20% of electrostatic, basically measures the energy between the molecule. The next term, the blue one here, is a dissolution energy. It's an empirical energy function, which basically measures the cost or the bonus of removing water from the interface between the molecules. So proteins are typically in water. So if you create an interface, you need to remove water. Maybe not fully, because there's still quite some water in between molecules in general, but you need to remove water. Now, if the surfaces of those proteins are hydrophobic, removing water is usually a good thing. So you gain energy by doing that. If you have high charges within the interface, then you have to pay a price because it costs energy to remove water from those charges. So this is what this term is basically measuring. And the last term in this very simple scoring function is basically measuring the amount of energy which is remaining based on the data that we put into the modeling. So if you satisfy all your data, this term will be 0. If you satisfy very poorly your data, here you would have a high contribution. And this is what we are still using. So this was developed 20 years ago. We have been revisiting this many times. We want to, students often want to come and do machine learning projects, so they train again models. But at the end of the day, this is a very robust function which still works very well for different types of molecules. It's also important to realize that when you optimize heavily scoring functions, they might work well for protein-protein and maybe even for a specific class of protein-protein complexes, but you cannot use these functions for other type of complexes, and this limits the applicability of those. So here we have a simple potential, maybe not optimal, but it does the job in most cases. Of course, we are also moving into artificial intelligence, and we have been developing AI-based scoring methods, first describing basically the interface of the complexes as a 3D image, and using image techniques, image processing techniques, artificial intelligence to recognize what will be a good model from a bad model. And then we move, so this was the DeepRank software, which was published first. And then we move more to the replacing a 3D representation of protein by a 2D graph, and graph has advantages. And here again, we train a deep neural network to basically classify those graphs as being a good model or a bad model. So that's DeepRank GNN. So it's a graph neural network, which was published last year. And one of the important features, what are going into these models when we train models, so the type of amino acid, the polarity, the amount of surface between the molecule, the charge, but also a very important component of those model is what is called the position-specific scoring matrices, which is a measure basically of conservation of amino acid at a given position. Now, the disadvantage with that is that, first, it's not going to work if you have molecules for which conservation is not playing a role in the interaction. So think of antibodies that are evolving extremely fast by definition. That's to adapt to new threats. And the second disadvantage is that you need to calculate those profiles from large sequence alignment, and this is taking time. So if you want to go very high throughput, this might be a limiting factor. Another thing that happened in the last years is the advent of language models. So Meta, Facebook came with the ESM2 language model, and these models were trained on billions and millions of sequences, and they were basically trained to predict which amino acid is at a given position in a sequence if given the context of the sequence. And this contains information which is also valuable and can be used in scoring purposes. So we replaced the PSSM profiles, which were more computationally demanding to calculate, with these ESM2 embeddings, which are very fast to compute. And you can also get them, of course, for other types of molecules like antibodies, nanobodies. And this leads to a new version of DeepRank GN and ESM, which is faster and performs as well, if not better, than the previous one. So that's something that in the future we hope to incorporate also in a Haddock pipeline as an option, additional option for scoring the complexes. So most of the users of our software are using it through a web portal, so a service that we're operating in Utrecht since 2008. So we have now more than 15 years of operations, and we are able to provide this service. So it's computationally quite demanding to run Haddock. You know, it's not, you don't want to use Haddock to screen millions of small molecules. But we can do that because we have access to very large computational resource distributed across Europe and provided through the EGI and the European Open Science Cloud. So currently, more than 80%, 85% of all the computing which is being done through our portal is happening somewhere in Europe. And even beyond Europe, some jobs are running in China, in Brazil, in the US. So we have served more than 640,000 docking runs since the early days. And the user community has been growing strongly. So this is the current world map of Haddock. So we cover quite a large fraction of the globe. And I checked yesterday, we had almost 900 users registered from Turkey, actually, which is very nice. We're still in a kind of an exponential growth in terms of number of users, which is also good news, because you might think that, you know, with all what is happening in the AI era and AlphaFold and these kind of methods, that maybe there is no need anymore for the more classical modeling approaches like the one that we have developed, but we don't see that happening yet. Maybe it will in the future, we don't know. Also, the number of new users that register every month is actually increasing. It almost doubled during COVID, so people realized, we cannot do experimental work, but we can use computational methods. So we had a lot of usage, increased users, and since COVID, this has been sustained, and it's still to be increasing again in the last year. So what can you do with that? I'm going to give you just two application examples, and then we'll go more into detail of some other applications. So the first one is the modeling of glycans binding to proteins, and this is a work that was done during COVID, actually in collaboration with Andy Baldwin and Benjamin Davis. They are both at Oxford, but there was a large group of people working on that, and our contribution here was basically to use NMR data that were measured for the ligands. So it's called saturation transfer NMR. And this was the work of Andy Baldwin. So they developed a very quantitative method to basically measure the impact of binding at an atomic level on the ligand. So this is information that we can give to ad hoc to model the complex. And the complex that we targeted was the spike protein of SARS-CoV-2 and the N-terminal domain, which had the early version of the virus had the capabilities to bind sialic acid in this region. So we generated a model of this interaction, which was later actually validated by cryo-EM. And this correlates also with the severity of the disease. Later on, the virus lost this capability to bind sialic acid. Some patients, some people have mutations in the enzymes that process glycans on the surface of cells, and as a result, they were exposing more sialic acid. And it was shown, it's also part of this story here, that actually these people got more severe long COVID effects because of that. The other story is it's another type of molecule. So one was, COVID. The other one is antibiotics, and that's the work of the group of Marcus Weingart, who is a colleague in Utrecht. Marcus is using solid-state NMR to study molecules that have antibiotic properties. And this is one of those molecules shown here. And one of the mode of action of this antibiotics-like molecule is that they bind to lipid-2. Lipid-2 is a important constituent of the cell wall of the membrane of bacteria. And those molecules are able to basically punch holes in the wall of bacteria. But since we are dealing with interactions at the membrane, it's very difficult to study by crystallography or cryo-electron microscopy, and NMRs, in particular solid-state NMR, is providing a lot of information. So using all the NMR information that has defined, which was defining basically the topology of the complex, how it assembled on the membrane, we used ad hoc to build basically a 3D model of the assembly of those molecules. So that's another very nice story where the modeling is, of course, a small part of the total, but everything together gives you nice stories. So I spoke mainly so far about ad hoc, which is our core project in my group, but over the years we have been developing many different types of software, some of which related to analysis of, say, cross-linking data from mass spectrometry, that's this. Some of them were more dealing with bioinformatic predictions, binding predictions, fitting the EM map at resolution. And the most recent addition is Arctic 3D. So most of these are provided as web services also from our website, NMR Science UNL. So we're not only providing ad hoc, but we're providing all kinds of tools around it, most of which are dealing with actually interactions between biomolecule. Now, Arctic 3D is the most recent addition to our battery of tools, online services, the work of Marco in the group. And basically what we want to do here is to data mine the protein database. At the European Protein Database site, so PDBE is standing for Europe, they develop what they call a knowledge base database. This aggregates all the information which is in the PDB and provides detailed information about all the interactions as well. So what Arctic 3D is doing is simply querying this database. Given the identity of a protein, like a UniProt ID, we're going to query the database, retrieve all the interactions, so all the complex that that particular protein does with anything that we can find in the PDB, analyze those complexes, define the interfaces, cluster the interfaces, and this ends up usually in defining a few binding surfaces. You can visualize those, but you can also extract the function by looking at the Go term, for example, and you see that in some cases, different interfaces are linked to different function of the protein. So this is all information, again, that you could feed back into Haddock to model interaction. So I think that's an interesting. So if you have your favorite protein that you are working on, it might be a good idea to just give it a try and see, okay, what kind of knowledge is there in a protein database and in other database about this particular protein. So now I'm going to show you a bit the future of where we are moving with Haddock and the software. And this is something that has been initiated quite some time ago, and different people worked on this project over the years. Rodrigo is still in the group, Brian and Joao, who left the group, and currently Marco and Victor are both actively involved in Haddock 3. So what is Haddock 3? So I explain you what Haddock does. We have these 3 stages, and it's basically in Haddock 2.x version. This is a static workflow. What you can do is tune the parameters, change some settings, but you are always running through this 3 steps workflow. So what we did in Haddock 3 is to basically break Haddock into small modules. And then you can mix and match those modules to generate new workflows. So you don't have anymore a static workflow, but we have a flexible workflow. And this allows us to do things that we could not do before. So where do we stand currently? So we have quite a number of modules that we have classified in different types. So the topology type 1 is what you need to basically build the system, build the description of the system. Then you will have sampling modules that are basically sampling this interaction space that I've been speaking about. With the rigid body one will be the classical rigid body docking in Haddock. We have different refinement modules, which allows you to basically optimize the complexes that you have generated. We have modules that are aimed mainly at scoring. So you have your model, you generated a model with AlphaFold, and you want to get some more classical energetics of the system, you can use those. We have a lot of analysis modules to calculate all kinds of properties of the system. So this facilitates our life a lot, because when you develop software, you have to do a lot of benchmarking, show that the software is working fine, develop recipes to apply to given type of problems, measure the quality of the models and all of that. So this modular version of Haddock really has a lot of advantages compared to the previous one. And it's quite simple to run. So currently we don't have a web interface for it, but the workflow that you define are simply defined by this is TOML formatted for those of you who are more maybe on the computer side of things. And the workflow is defined here. The red part defines the step of the workflow. And you only need to define the parameters that you want to change. Everything else is done by default. We are also built into Haddock 3 capabilities to generate statistics, to generate plots. Those plots show you, for example, the distribution of the different clusters, their scores. This is something which in Haddock 2 is only available from the web server. So if you run Haddock 2 locally, you have to manually generate all of this information. While now, if you run Haddock 3 locally, you will get all this information generated for you. So that's also a very nice advantage. And we are working on building a web application that will basically integrate Haddock 3 and offers the ease of use that we have with the Haddock 2 series. So this is very much work in progress. So now it's time to move to some real applications. So what can we do with all of that? And I will start with antibodies. So antibodies consist of a heavy and light chain. And in antibodies, you usually know where the binding takes place. These are in these hypervariable loops that are at the tip of the heavy and light chain domains. So in these regions, they are defined. So there are 3 loops in the heavy chains, 3 loops in the light chains. And these are what are called the hypervariable loops. These are very quickly changing in our pool of antibodies that we have in our body. And these are maturing basically in a reaction to infection, bacterial infection, viruses, whatsoever. So usually for antibodies, we know where the action is, when you know where the binding site is. And this, for Haddock, is a perfect information. So we know on the antibody where things are binding. So in the past, we've worked on antibody modeling using more a classical approach where we say, okay, we have information on the antibody, we have some information on the epitope. So the epitope is the binding site on the antigen, so the target of the antibody. And we've been testing different scenarios and testing different software, comparing different software that can use to some extent information to guide the modeling process, and showing that Haddock was doing a really good job. Now, we are in this AI era, and before that, we were basically limited to working on systems for which there was an experimental structure of the components. Excuse me. Now, these days, with all the AI development, we can take sequences, basically, and from sequences start generating structure. So meaning that we don't need to have any more structures of antibodies if you want to start modeling with antibodies, but just knowing the sequence is enough to start the modeling process. So a number of tools have been developed in the last couple of years that are dedicated to modeling antibodies. So Immune Builder, that's from Charlotte Steen's group, is an AI model. In the meantime, so you see a bioarchive reference here, but it has been published. The Rosetta family in a group, basically the group of Jeff Gray has developed IgFold, which is also a deep learning model to generate antibody structure. AB Looper was another model from Charlotte Steen, and then we had AlphaFold, of course, which can deal with antibodies. So these are 4 tools that are easy to use, and which you can with which you can generate from sequence antibody structure. So what we wanted to do here is basically test the quality of those tools to generate antibody structure, and then use these antibody structure, or models, I should call, to start modeling antibody-antigen complexes. So we're not going to do a modeling in this case without any knowledge on the antigen side. So what we are aiming at here will be more like an antibody design pipeline, where we say, I have a protein, I want an antibody to bind at a given specific region, and then I want to optimize this antibody. So we are very targeted modeling question here. So this is, we have a collaboration with a company, actually, Exscientia, where you see Constantin, Nikita, and Danielle are working at Exscientia, and Marco is working in my group. So what is important, and this is important for any application that you're going to do using AI. So if you want to test the capability of AI model to do prediction, you should make sure that the system that you are using for this testing have never been seen by the model. Because if the model have been trained on the same sequences, then it's much easier for it to predict the right structure. So, and this is not always happening these days. So if, if structures are not properly or are part to some extent in a training set, then you are over-representing the performance of all these AI models. So we made sure that no structure were present in the training set of any of the method that we are testing. And for that, we check the sequence identity, but not only the sequence identity of the entire sequence, but also of the hypervariable loops of the antibodies, because these are the ones where the binding takes place. So we could come with about 70 unique antibody-antigen complexes, which we could use as a really independent test of the methodology that we are developing. So here is the results. So using the different method that I shortly presented, so we have AB Looper, Immune Builder, AlphaFold, IgFold. So you see here a measure of the quality in terms of root mean square deviation. So the deviation between the position of the predicted loops with respect to the position of the loops in a crystal structure. So we know the answer in this case. And you see that all the methods are doing quite well. The median RMSD is around 1.4 angstrom. And what is also interesting to see is that if you start using mixing models, so if you mix all the methods that you have, you can improve actually the modeling around to about 1 angstrom. So in general, all the methods are doing quite fine. Of course, there are always outliers. Now, if you know antibodies, you also know that there is one loop which is particularly challenging to model. It's the H3 loop on the heavy chain, and it's more challenging to model because this loop is usually longer than the others. And the longer the loop, the more tricky it becomes to generate accurate conformation. And this is represented here. When you measure over the entire binding interface, you see that we are at 1.41 angstrom accuracy. If you measure only on the CDR-H3 loop, you see that the accuracy, that this is the median, is above 2 angstrom, and you see that we have outliers that are up to 10 angstrom. Okay, so there we will have problems with those models that are 10 angstrom away because the docking method will not be working very well. And again, here the ensemble approach works best. Now, if you look at using AlphaFold in this case to predict the structure of the antigen, this is working, you could say, perfectly. So we should not worry too much, although there are always exceptions, and you see in some cases there are some models that are going to be very far away from the true answer. So we have all these methods, we have all those models, so now we need to put them together. And we're going to use haddock for that, given the knowledge of the hypervariable loops. So we have two basically scenarios. In one scenario, we say, okay, since it's what I was explaining to you more, design scenario, we want to target the binding sites. So we take the knowledge of the binding sites in the complexes to target the docking. This is all kind of cheating, so it's going to be easier. A more realistic one is to say we know the hypervariable loops on the antibody, so we're going to use all this information. And we have some more vague definition of the binding site on the antigen. And we're going to test that for the antibody. We're always testing basically, so we're going to generate the models from the different AI methods. And for the antigen, we are comparing both using the crystal structure or the prediction. And in some cases, this is going to work very well because the prediction is on top of the experimental structure. In some cases, here you see in RMSD 11 angstrom, you're going to run into problems because the model that you generate with AlphaFold is quite far from the true structure, and of course, this is never going to give a very good docking model. So now we used haddock-free, and we have different recipes. So the first one is say, okay, let's take one model at a time, and then we do a very short sampling. I was telling you we sampled thousands of 10,000s of model. Now we only do 48 docking runs here, and we add flexibility at the interface and final minimization. In the second workflow, we're going to use an ensemble. So we combine predictions from different AI methods. We increase the sampling to have the same number of samples per model that we give. We select the best model at this stage, 48. So we do a ranking step and we refine it. And in the last one, we do the same, but instead of doing a selection, we might lose good models because at this stage, the models are not yet refined. We cluster. This was not possible in haddock 2. We select from each cluster 4 models, and then we refine everything. So first of all, of course, we tested the capability of AlphaFold multimer in that case to predict those complexes. And you can see the accuracy so is quite limited. So if we say take the best model generated by AlphaFold, our success rate is only 18%. So for only 18% of all the complexes that we have to predict, there is actually an acceptable solution. If you look at the best 10 model, it's 22%, but it's not fantastic. AlphaFold 3 claims that they are better at this. They have improved things. Now, here are the results of the modeling using haddock-free. So this is, let's concentrate mainly on the right side. So this is the more realistic side where we have the knowledge of the hypervariable loops and the vague definition of the epitope. What you see on the y-axis is the success rate from 0 to 1. So if 1, it means everything was predicted correctly. If you have 50, it means that in only 50% of the complexes that we are predicting, we got a good solution. The color on the x-axis, you see the different AI models that we are using. So these are the different models, and these are combination of models from different AI methods. Red is just testing the ability of haddock to rebuild the complex. You take the crystal structure, you take it apart, and you redock it. No conformational changes, so that's the ideal scenario. And you see that here we are doing very well, but that's not a realistic scenario. It just defined the baseline. What is the best that you can do if there was no conformational changes in your system? And then we have the blue color, we use the crystal structure of the antigen, and more the orange color, we use the AlphaFold prediction of the antigen. And of course, here you see that our performance is around 60 to 70%. Some more general conclusions here. So first of all, Immune Builder is one of the AI methods that gives you the best models. And that's nice because Immune Builder runs in seconds, while AlphaFold will require GPUs and take tens of minutes to make a prediction. So the fast method works very well. That's nice. The other conclusion that we can draw from these results is that if you combine models from different approaches, actually you and use the ensemble docking capabilities of ad hoc, you are doing a better job. So we get a higher success rate. So what is the limiting factor here? So in our ability to get good prediction. So what you see here on the y-axis is a measure of the quality of the model that we are generating. It's called the DocQ metric. If it's 1, it means you have the perfect model. If it's 0, it means it's completely crap, completely wrong. And what you see on the x-axis is basically the quality of the prediction of the binding site of the paratope, so the loop, the hypervariable loops on the antibody. And you see that as the prediction becomes worse in general, the quality of the docking model becomes worse as well. So it's not a perfect linear correlation, but you see there is a trend. And this is basically what is happening. So it would be nice if you were to be able to predict the quality of the prediction, the AI prediction, to say, oh, this was a good loop prediction or not, because then you know where you are in this graph. But this is very difficult to do. So in conclusion, for antibodies, so the tools that we have at hand, AlphaFold and the other tools that are being developed, and there are new tools coming out probably every week. bioRxiv is flooded with AI papers. They are very accurate in modeling antibodies, and they generate conformation that are actually quite close to the bond form, so the form that you need to bind to the targets. We can, using those models and using haddock-free, the new version, and a very limited sampling protocol, we can get very good performance. And the key in improving the results is basically to use ensemble of models. But of course, there's still room for improving. The scoring remains a very difficult thing in general in the modeling of interactions. So now time is going on, and I don't want to take too much of your time, so I'm going to go very quickly through this one. Just to show you that haddock has been used in the context of small molecule. These are 2 publications that were published by pharma companies actually using haddock. And the reason they went to haddock is that they had experimental data, they had distances, but the classical docking software, small molecule docking software, could not use this information. And this was key to getting good models and doing drug design based on the model. We have been participating to competitions. There was these grand challenges, and we learned a lot about small molecule docking, not doing very well in the early rounds, and then improving our capabilities. And the latest protocol that we developed for doing small molecule docking is kind of a template-based protocol, and you see we call it shape-based protocol, so it's Manon and Panos work. But basically, for many drug targets, you will find in the PDB some structure with some other small molecule bound to it. Now, if you can find this information, so unless you're working on a completely new drug target and there's no structure at all, but in many cases, there is quite some structural information in the PDB, you can extract the ligand that resemble most to the ligand that you want to model, define this ligand as a 3D shape. So these are fake beads, fake atoms. They have no energetics with the remaining of the protein. And then we define distance restraints from the ligand to those beads that are basically telling the ligand you should be overlapping somewhere with this shape that we define. And this is a very efficient protocol to model small molecules. So I'm going to skip all of that and just show you the performance. So again, we have a benchmark set, about 100 complexes. This is rigid body docking. This is still done in haddock 2, this one, but it works also fine in haddock 3. And after flexibility, you see that this is the number of models that you consider. So if you take the top 1 model, we reach a success rate around 70%. You go to the top 10 model, we reach about 80%. The difference between the shape and the pharmacophore model, if I go back here, in a pharmacophore model, you might derive a pharmacophore model of your small molecule, and then you know that in some regions you should have aromatic groups, in some regions you might need positive groups, in some region negative group. So you add a flavor to the beads that we are using. So that's the difference between the two. But this template-based shape restraint protocol works really extremely well. And what is also striking of this approach is that you can induce quite large conformational changes in the ligand. So you see here 3 examples with conformational changes of more than 4 angstrom, where in white and orange you find the crystal structure of the complex, and in orange will be the haddock model, and in cyan colors was the starting points before the flexible refinement. And the shape is really driving conformational changes. That's quite an advantage. We also know when to use this protocol or not, because you can measure the similarity of your ligands to the target, to the template that you find. And if you have a high similarity, say above 0.4, then you see that the protocol works very well. If you are below that, you are in the problematic area. So we have defined kind of a safe similarity performance to use this protocol for ad hoc, and it has already demonstrated quite some applications in our hands. So it's also competitive with our software. So this is a comparison of the performance of our shape-based protocol. Green, higher is better in all of these. What I should say is that here we did the docking by starting from the free form of the proteins, while all the other testing for the other software, which are classical small molecule docking software, was done from the crystal form in the complex. So there are no conformational changes on the protein side for the assessment of the other software, while in our case, we also need to deal with conformational changes on the protein side. So we are basically underestimating our performance here, or maybe we are overestimating the performance of the other docking software. Okay, so to finish, I'm very proud that we have developed a software now for more than 20 years. It's still alive, it's still moving forward, it's still useful to the community. So it's not an easy thing to do, and that's especially in this area where AI, everything becomes AI. Things are moving extremely fast. It's good to see that we still are in need of more classical modeling approach like ad hoc and some other ones. So the good old energetics, the good old physics is still worth something. And we are moving now toward this very flexible and customizable workflow with ad hoc 3. This is the current team of people in the lab in Utrecht. Funding from a large number of projects over the years, the Dutch Research Council, but also European funding has been very important in further developing the software. With BioExcel, the Center of Excellence for Computational Biomolecular Research, funding further development of ad hoc. And also the EGI and European Science Cloud providing all the computational resources so that you can use the tools freely. And this was a picture of our 20 years reunions with most of the contributors to ad hoc over the years present. With that, I thank you for your attention. I will be glad to answer any question if you have any. And you can find information about ad hoc and the software, and also a forum where to ask questions online. Thank you.
Thank you, Professor, for your valuable and precious talk. Now we can start our question and answer session. There is a question. Would you like to read it, or I can read it?
Maybe you can read it, or let me see. I can turn on the chat. I'm still sharing my screen. Okay.
Okay.
Oh, let's see. Okay, AI will shorten the range of treatment in cancer. Okay, so that's a question that's not I, but the field gets very often. I think AI is another tool in the battery of tools that we have in structural biology. I don't think that you will see on the short term a drug developed purely on AI without any experimental validation. It still takes a long time. So by the time you